On behalf of National Institute of Plant Genome Research, it gives me immense pleasure to welcome all our guests, staff, and students present both here and online on National Technology Day. I extend a warm welcome to our chief guest, Professor Padmanabhan Balaram, and to Professor Hemlata Balaram. On the same note, I welcome our guest of honor, Dr. Rajesh Gokhale, Secretary, Department of Biotechnology, and I request you, sir, along with Professor Balaram, our Director, Dr. Shubhra Chakrabarti, and Dr. Alok Sinha, Scientist, NIPGR, to grace the dais. I invite the dignitaries on the dais to begin today's function by auspicious lamp lighting ceremony. Dr. Shubhra Chakrabarti will now deliver the opening remarks. Okay, prior to that, I request Dr. Shubhra Chakrabarti, Director NIPGR, to honor our dignitaries. So now is the time when our director will deliver the opening remarks.
A very good morning to one and all who are with us in this auditorium as well as online. And I welcome all of you to this Technology Day event at NIPGR today. And we are very fortunate to have Professor Balaram from who I think doesn't need any introduction to any of us in Indian science, and to kindly agreed to deliver this Technology Day talk today to us. A little bit, and also we are very, fortunate to have our Secretary Deputy DG Brick, as well as our Chairperson Governing Body NIPG are with us today morning. Somehow some meeting got cancelled, and we are lucky to have him with us today. And we have many of our directors from NCR region of DBT institution, many other colleagues, and also we are lucky to have Professor Hemlata Balaram from JNCSR, who also kindly agreed to come and visit NIPGR and to be with us on this day. I'll not be standing between you and Secretary DBT and Professor Balaram. Just two few words that India celebrate this technology day, which all of us know from 1998 with the triumph of the Pokhran that happened that time. And similarly, this year also India is celebrating the technology day today. And you know that if I look into the biological science, always biology has evolved through technology, and starting from Mendel's discovery, and now today almost going to be more than 270 years of the biology that we have seen. Only one thing that I feel possibly initially the biology was driven by more of inquisitiveness and kind of, all discovery came from some observations. And starting from the Mendel, little by little over the years, it has changed. And after the discovery of the double helix possibly, I think we have gone into more of technology. And now we cannot think biology without technology. So that is where we are today. And this institute started with a mandate of doing, discovery research, and we use technology to understand many of the unknown and to question many of the unknown in the area of plant biology, and to understand how plant cope up with different environmental challenge, how plant develop, how plant can make more nutrition, and so on and so forth. But always we actually utilize the technology to understand those and ask those questions to make hypothesis-driven discovery work, and any of our such work gets translated as a biotechnology using many of the biotechnological tools like genome editing, genetic engineering, so on and so forth. So right now we are with the amalgamation of both biology and technology, but I think we should have more of basic research, and then only we can have more of, translation that we want to do it for societal benefit. So with that little bit, I'm really very happy, and I again welcome Professor Balaram, Dr. Gokhale, Professor Hemlata Balaram, many of our colleagues from DBT NCR Institute, and all of you, and the NIPGR family, and we look forward to the talk and always it would be a thought-provoking talk that Professor Balaram makes, and we learn so much from his saying. And also we look forward to the address by our chief guest, Dr. Gokhale, and very dear and near to us as Secretary DBT, and more of that, I would say, Chairperson, Governing Body, NIPGR. With that, thank you all.
It is my privilege to invite Dr. Rajesh Gokhale to grace us with his enlightening words.
Namaskar and good morning to all of you. I think Professor Balaram was going to come here. So Dr. Shubhra mentioned to me in about a day back. And, I was just wondering, I was going to miss him actually this time. When he comes to Delhi very often, but when you want to meet him, actually, it's always being my mentor for decades actually now. And all of a sudden, the Prime Minister's office decided to cancel the meeting actually and postpone it to Tuesday. So it turned out really well for me. And so here I am today actually amongst you to listen to his wonderful stories that he actually gives around with his English humor. It is always very, very interesting to hear from various aspects of life that he has been over the years done it. I think this institution means a lot to Department of Biotechnology, actually. And I must say this, that this is one of the premier institutions in where plant biotechnology actually has in fact been progressing, and in fact has given out to entire countries progress, good science, great technologies, great great environment. And I think we are all really, really very happy that the Technology Day celebration, actually, that many of my colleagues from DBT, directors and other institution leaders are here, that we all gathered together to discuss something that how the path for future of the country can be decided about. And I think the reason why I think this is because just the way the IT industry actually changed the world in 1990s, the biology is going to be the next technology revolution for the next 2 decades to come. And very often the responsibility lies on the early movers of how to really make that happen, how these contours can be placed together and how can we really put the early bricks such that these actually then develop into a very big systems. Often it is said that the IT industry grew only because government never knew about it actually. And if government would have known it, they would have realized they would have done enough to make sure that the IT industry would have suffered. I don't know if that is possible in today's world and era where the connectivity has become social media connections, news media, everybody's intervention is so much that the technological revolution is not going to happen in a silent mode, what we have seen in the past. I think that is where the responsibility lies with government, with the organizations, with institutions, and primarily with the institutions actually, because I think that's where all the good things happen actually. And that the government is really a catalyst in its own ways, in the way that it kind of, chaperones activities and provides incentivizations and mechanisms to be able to do. But I think increasingly the participation between various aspects of our lives are becoming more and more critical. And I think one of the most critical and important thing that will happen is the younger generation's participation in what government is talking about Viksit Yatra actually, primarily because the speed with which science and technology is changing is actually far faster than what I would have thought and realized when I was maybe doing my, at an early part of my career, when I started my career 2 decades back. I wish I was young enough to start my lab again. Sometimes that's what one of the things I miss, because there's amazing biology that one can do right now today that we only dreamt about at that time actually. And so it's actually great opportunity for all of you. And you can see the Technology Day mission as well. I think the one that's announced by is also from schools to startups actually, because the real challenge is how do you build innovation? How do you take it to the next level of activity? And it's unlikely to happen in a very classical model because obviously there are many other challenges that you face, which involves financial capital, which involves getting right kind of people together, having support from government to be able to look at. So I think that from the Department of Biotechnology, we've been trying to do our best to create new ecosystem, create new plans and ideas. And our future, as we've been talking about biomanufacturing as one of the perspective of, it's basically a movement for us to drive to say, can engineering biology be a real success stories for future to come? And that's in a various domain sectors. That's what we are talking about. It's not just related to, one aspect of it. But you'll be amazed how wonderful these things can be done in a very, very short period of time. For example, I don't know how many of you know this, but in our country, the chemical industry, in fact, produces about 70,000 to 80,000 molecules. And many of these molecules are derived from oil to chemical industry, actually. And I think the precursors come from what they call O2C market, actually. And whereas what we want to look at is, can the B2C market actually become a reality in times to come? Now, how would you make it happen, and how do you really create these kind of challenges and ideas and solutions for future to come? Is really going to be an exciting journey. And you can clearly see that the data science that is coming out of biological systems based methods and ideas is an incredibly powerful tool for you to actually come up with plans and ideas that are completely crazy and can be transformative in the future perspective to look at actually. So I think that's why I think that the younger generation really has to grow up very, very quickly. And as we have been talking about a PhD program that we are discussing through BRICS, which we call I3C, the whole purpose of that PhD program is to be able to tell PhD students that PhD, you should take it as part of your first job, rather than looking at as a training for yourself to be able to look at. Because many of you come very well trained and through various institutions that you have already spent your time and have already done several internships, several, lab work, several schools, several kinds of courses and structures you've already looked at. So I think it's a tremendous opportunity for us to start looking at in a very different mode. I mean, the knowledge is available almost, the information is available everywhere. You only need to learn to figure out and synthesize knowledge from it and how to be able to do this. And I think this is a great opportunity for us. And as you would realize, that Department of Biotechnology from various aspects is tremendously supporting all these initiatives. And several directors from our DBT institutions are here. It's very good to see them from NCR. Actually, I think we are all, the whole purpose of doing together is that many of our institutions have been working and doing fantastic stuff, but it's been going on in silos actually. And now with an integrated impact, about 400 scientists in Biotechnology Research Innovation Council does becomes the biggest body that it will actually train and create people and create scientific enhancement and take bold decisions and actions that can determine the country's progress and future actually. And from that point of view, integration is a really, really exciting opportunity for us to work together, bring together our best in different ways and ideas, and I think come up with a plan that are much larger than what one can dream about actually. I think I'll stop here. We all look forward to Professor Balram's lecture. As I said, I have grown up listening to him since I was, of course, student in his lab and was been my mentor there. And I think it's always exciting to hear wonderful things. I also thank Dr. Hema Balram for being here. It's wonderful to see her after a long time. And thank you all of you being here. Namaskar.
Our chief guest, Professor Padmanabhan Balaram, is a senior visiting professor, NCBS Bangalore, and chairperson of Ashoka Trust for Research in Ecology and Environment. Professor Balaram did his BSc from Pune University, MSc from IIT Kanpur, and PhD in chemistry from the Carnegie Mellon University, USA. His postdoctoral training was at Harvard University. He has served as a faculty at the Molecular Biophysics Unit, Indian Institute of Science, Bangalore, from 1973 to 2014. Professor Balaram has also served as the director of ISC from 2005 to 2014. His research contributions have been in the areas of molecular biophysics and chemical biology. Professor Balaram has been honored with Padma Shri in 2002 and Padma Bhushan in 2014. The American Peptide Society conferred him with the R. Bruce Merrifield Award in 2021. He has authored more than 400 research papers and is a fellow of the Indian National Science Academy. He has been the editor of the journal Current Science till 2013. It is my honor to invite Professor Balaram to deliver today's lecture on nature's technologies, the evolution of biochemistry, and the birth of biology.
Please, sir. You don't have. Can I speak first? Will that work? Or give me that.
Has but moved.
Come in the corner. Okay, good morning. I don't know whether to use this one or this one. Maybe I'll use this one. Right? I usually prefer a collar mic so that I can walk around. But the collar mic that you have is the one which has that thing around the ears. And that makes you feel terribly constrained. Anyway, good morning. And I must first begin with an apology. And the apology is that I'm the wrong person to be talking on National Technology Day. I'm not really a technologist. And if I were to describe myself, I would describe myself really as a student and a teacher of science. So that's all that I've been throughout my career. And when I hear the words science and technology, I'm always convinced that science and technology are two sides of the same coin. You can't have technology if you don't have science. You might desire many things, but you can't achieve them unless you really understand what you're doing. Now, we celebrate 2 days, a National Science Day on the 28th of February, which celebrates Raman's announcement of a new form of light scattering, later called the Raman effect, on the 28th of February 1928. Now, when Raman really discovered the Raman effect, it had no application whatsoever. It received a Nobel Prize 2 years later, only because it was, I think, the first real demonstration, experimental demonstration, of the validity of quantum mechanics, the quantization of energy levels, and in this case, vibrational levels in molecules. Today, the Raman effect is used everywhere. It is a technology. And just this morning at the CSIR Science Center, there was a young scientist who was discussing with me the possibilities of using Raman spectroscopy to try and distinguish between, in Pashmina shawls, the introduction of fibers from a rare endangered species of antelope, Himalayan antelope, the chartreuse. And this is a problem which is, I think, a concerning problem. National Technology Day is on the 11th of May. That's today. And what this celebrates is the 1998 Pokhran 2 explosion. And so I thought I would also remind you that a week later, we will actually be marking the 50th anniversary of the Pokhran 1 explosion, which was the first time that a nuclear device was actually exploded on Indian soil. So we've chosen the nuclear explosion as an example of a technology. And it is an example of a very powerful technology which comes straight from atomic physics and which is sort of now really immortalized in the history of science. Who are the players here in making the advances in atomic physics which began? You remember that the 20th century really began with Max Planck, Albert Einstein, and the quantum. Planck introduced the idea of the quantum. Einstein established it with the photoelectric effect. How did the 20th century end? The 20th century ended with a picture of Francis Collins and Craig Venter flanking President Bill Clinton when they announced the first draft human genome. So now, of course, I'm in the center of plant genomics. We've come a long way from the year 2000 to today. These were all large projects. They were projects which demonstrated technologies which were possible. But what they needed really was, and what government today calls mission mode projects, we can take a lesson from the atomic bomb project. There were two people here whom I've labeled there as the unlikely pair. This picture is from the Nuclear Museum, which is available on the internet. And there you see Oppenheimer and General Graves. There was the project manager, and there was the scientist who could understand the science of all the other scientists who were working on the Manhattan Project and bring the technology of a nuclear explosion to completion. Of course, today, I haven't seen it, but many of you would undoubtedly have seen the movie Oppenheimer. And there you can see that the recreation of the individuals is quite remarkable. But when we talk about science and technology, and I'm going to come to the subject of my talk in a moment, but I thought I would start a little bit generally because it's National Technology Day. I must show you this book called Pasteur's Quadrant, Basic Science and Technological Innovation. This was written by a professor of political science. Why a professor of political science? Because it turns out that science and technology are very important in politics. In India, they're not. In India, the only word that is used by politicians is DNA. And every other day, I see that some politician or the other says something is in their DNA or something is in somebody else's DNA. And Donald Stokes died before he published the book. And I say this because the picture of Pasteur's Quadrant, which appears in his book, is a black and white picture with only a few things written on it. And that's what I will show you on the next slide. But the next slide is Pasteur's Quadrant, as I have redrawn it, now, of course, taking liberal use of Google Images. So I've illustrated each of the quadrants. What are the two axes? The two axes are use-inspired research along the x-axis and fundamental research along the y-axis. These are Stokes terminologies. Today, I could use translational research along the x-axis and basic research along the y-axis. Those are terms that we understand. Today, there is no term which worries me more than the word translation. And it's sort of ironic that I was asked some time ago by CSIR to go and give a lecture on translational research in a series which was called the Amrit Kal Lecture. Now there I did reflect on the fact that we are misusing the term translation quite a lot. Now use-inspired research is best illustrated by Edison. Edison could have discovered the electron, and that's what Stokes says. In his book, there are notes, in his notebook, there are notes which say that Edison was on his way to discovering the electron. But Edison was not interested in pursuing that discovery further. What he was interested in doing was in creating illumination or in inventing the light bulb. In that process, of course, Edison became an inventor, a technologist, and also one who illuminated our lives. Along the fundamental research axis, the top left quadrant is called the Bohr quadrant. These are designations given by Stokes. I've only illustrated them. Bohr gave us the first model of atomic structure. It wasn't the right model of atomic structure, but it was the one which really catalyzed the quantum revolution in physics. And our entire understanding of physics afterwards is really catalyzed by Bohr's initial efforts to explain the atomic spectra of hydrogen. On the top right, which is the quadrant where we would all like to be, is called the Pasteur quadrant. And that's what he uses in the title of his book. This is because Pasteur is a man who exemplified two things. He's like Faraday, both the highest quality of applied research and the highest quality of fundamental research. Pasteur, of course, is the man who resolved the tartaric acids, who introduced the first ideas of organic stereochemistry long before anybody had an idea of what the structures of molecules were. And he's in many ways the founding father of three-dimensional structures and their application in biochemistry and in biology. He also, of course, gave us the most fundamental work in microbiology and also the most applied work in microbiology. And today, when we talk about vaccines, when we honor him with the term pasteurization, we're really paying a tribute to a man who did the highest level of applied research. That's the quadrant into which we would all like to be. So on National Technology Day, you might well reflect, in this space of science and technology, how do institutions and individuals navigate this space? Where do we all begin? We begin at the origin. Everything begins at the origin. Right at the beginning, we are neither scientists nor technologists. We know neither science nor technology. We study, we learn, we do research, we improve, we evolve. Stokes did not illustrate the bottom left quadrant. And I was looking for an image. And I thought no image would do better than Lakshman's common man. Lakshman was a cartoonist. For those of you who are too young, an immortal cartoonist. He would have loved today's politics and we would have had a wonderful cartoon every day. But we don't have Lakshman anymore. And that's where average academic and industrial R&D is. And we're all trying to search for our own way on how to get from the bottom left quadrant. It doesn't matter if we go to the top left quadrant or we go to the bottom right quadrant. Both of them are wonderful quadrants. But we now have organizations and people who are somewhere lost in the middle. And we have to find a pathway. And if you know your chemistry, if you want to go from molecule A to molecule B, there's a large activation barrier to be crossed. So even in academic space, if you want to go from point A to point B, you must cross this activation barrier. But before I come to the real subject matter of my talk, which is the evolution of biochemistry, I must tell you that biology has made a huge impact on the public imagination. And nothing has made a greater impact on the public imagination than the pandemic. What is wonderful is that we've forgotten the pandemic so quickly. And what are the lessons that we've learned from the pandemic? The first lesson is that nature periodically provides a reminder of the limits of human arrogance. This is the first lesson that you must learn. The second is that nature demonstrates that the frontiers of science are truly endless. And last of all, as I will show you in the next few slides, the pandemic also demonstrates that biology, assisted by the chemistry of molecular variation, is a formidable force of nature. I've illustrated this slide with 4 images. The virus, of course, all of you recognize. On the top right, you have a slightly more expanded view of the virus. It's an enveloped virus with a lipid membrane into which a protein is embedded. But the spike protein is shown in greater detail at the bottom here. What you get from cryo-electron microscopy. But on the bottom left is the classic image by which the coronaviruses were discovered in the mid-1960s. This is the famous picture which David Tyrrell and June Almeida took of a sample provided to them by the American microbiologist Dorothy Hamrick. What's the definition of a virus? This is the best definition. I'm not a virologist. I got this from Peter Medawar's book, A Philosophical Dictionary of Biology. The title of the book is Aristotle to Zoos, A to Z. And he says, viruses are a piece of bad news wrapped up in protein, which is true. The bad news is the nucleic acid, it always is. And the protein, of course, manages to help in getting the nucleic acid into host cells. What is the burden of the pandemic? We've forgotten it so quickly that the Johns Hopkins site, which was maintaining all these statistics, ran from the 22nd of January 2020 all the way to the 10th of March 2023. And all you need to look is at the figure of deaths. It's 6.8 million here. You can multiply it by a factor of about 2.5 to realize what the actual worldwide deaths were during the pandemic, which would be about 15 million. We pay so much attention to many, many other diseases where the death toll over a much longer span of time is much less. And here was an event which happened in your lifetime and in mine, in which in a period of 2 years, the world came to an absolute standstill. This is a graph which I've just made. Might show you why. This is the evolution of the surface protein of the coronavirus. And this is the spike glycoprotein, and this goes all the way now till April 2024. This is data till the 20th of April 2024, because NCBI still gets sequences deposited. They have collection dates realized on them. And what is happening here is that you can now follow evolution of this protein over a period of 4 years. From the beginning of January 2020, along the x-axis, it's days from January, and you could see the viral variants coming. What I have on top of each one of the viral variants is the number of mutations that they have. And what you have along the y-axis is the average number of mutations. This is evolution happening in real time. We have had no other case like this in human history. Because before this, human beings did not have the technology to sequence. And before this, we didn't have a pandemic after sequencing came along. And therefore, this is evolution in action. And viruses multiply so quickly that we are going over a pretty long time scale over here in 4 years. You can see we started with one mutation which drove the pandemic, a single aspartic acid going to glycine. That is the D614G mutation. The virus, however it was created, whether it came by zoonotic transfer from bats or whether it was an engineered virus in a Wuhan laboratory, it does not matter. Once it is released, it's now evolving in a human host. And it's evolving under the stresses which the human host imposes on the virus. Can't get a better example. This is actually an experiment. When I say nature's technologies, this is an experiment that nature has conducted in basic science. It's let loose a virus and then asked the question, how would it expand how would it evolve in the human population? How will the sequences evolve? That's evolution. Never forget that word if you're a biologist, and it's a word that you must remember even if you're a generally educated person. Think about this a little bit. We had one mutation. Then at the height of the pandemic, when people were dying, that was 2021 summer, 10 mutations. We were rescued by the Omicron, 34 mutations. Moved on to 40, you can see that. And now the JN1 version, which is circulating, has 60 mutations. Now you can see that if I keep on going, if I keep on doing the sequencing, maybe after 5 years, maybe after 10 years, I would have 300 or 400 mutations in this viral sequence. And by this time, if I were now doing a phylogenetic tree, I would actually now give a different name to this virus altogether and say it has become yet another virus. And this is how things actually evolve. Think of the coronavirus pandemic as an experiment done by nature. It's an animal experiment. You've injected a virus into an animal, a human being. Afterwards, this is the only experiment that I know in biology where the animals are doing the analysis of the experiments. In all other cases, that is not possible. Now you can have, instead of looking at all the viral variants which the virologists have been doing, you can look at single mutations which are characteristic of each variant, and then over the period of days, from January 1st of 2020, you can follow this. And you can see the sites at which this is happening. You can also ask the question, how does this improve the fitness of the virus? Evolutions, after all, improve the fitness of an organism. Here I show you 3 sites which you might have heard of. The furin cleavage site, which is the one which led to the conspiracy theories on the engineered virus. The immune escape mutants, where you keep on creating. And here, since I am on a technology day, remember, you must ask the questions. Did the vaccines help or did they not? Did nature help us or did the vaccines help us? Today, when AstraZeneca withdraws its Covishield vaccine, saying it's of no longer any use against the variants, are the companies going to have vaccines which are going to be effective under the other variants? Or has a natural biological process now happened where the virus has no intention of killing the host, the virus and the host have reached an evolutionary equilibrium? We've had past encounters with viruses, and those encounters are actually embedded in your genome and in my mind. There are many things we can look at in this protein and with the data that we have now generated. Virus cell fusion is the first step to infection. The virus must recognize the cell, the membranes must fuse, and the RNA must get inside. And this has been known for years, since the classic work of Don Wiley on influenza. And here is Steve Harrison's summary of this sometime about 10, 15 years ago. But today, when you look at this, that if I can click off somehow, because that's my favorite picture. One of the things with these computers, they keep putting up something or the other which you don't want. Yeah. I've entitled this site as Viral Host Fusion and Wonderland. Wonderland, of course, is the wonderland which Alice found when she went down along with the white rabbit down the rabbit hole. And you can see that's the spike glycoprotein, 2 domains, S1 and S2. There has to be a proteolytic cleavage to remove the top red cap and then expose this. Then there will be this large conformational change of the protein, first observed by Don Wiley with influenza hemagglutinin. And you can see from 86 angstrom, the fusion competent protein now goes to 210 angstrom. This is technology. You've taken a material, 86 angstrom, you've now made it 210 angstrom, punches a hole in the host membrane, and that's how the 2 membranes really fuse. I won't go into the details of fusion, but this is a remarkable example. There's an analogy used in the literature, a metaphor. This is called the loaded spring model. So you have a spring, you compress it, and then when you compress it and you remove the compression, out comes the spring. Jack in the box. Reminded me immediately when I saw these structures that it's like Alice. You know, in Alice in Wonderland, she'll drink something, she'll become very tall. She'll drink something else, she'll become very short. And there it is, Alice going up and down, the viral fusion protein going up and down. And that's how all the enveloped viruses work. Every enveloped virus, whether it's HIV, influenza, the coronavirus, the Ebola virus, all of them work by this mechanism. You can actually now ask an evolutionary question, where did the viruses come from? The viruses must have also had an ancestor. And that's where evolution becomes very important. The science of the pandemic is important, remember that. And the illustrations on the corners of the slide only tell you how much the pandemic and the coronavirus and the spike protein captured everybody's imagination. From Chennai and Secunderabad all the way to France and Italy, these images appeared. I collected them in the months of March, April, particularly April of 2020 and May of 2020, when we had our first lockdown and we were stuck at home. But I'll remind you that when the HIV virus came out of nature, came out of nowhere, Lederberg, at the height of the HIV problem in 1988, wrote this. Joshua Lederberg, of course, is one of the immortal figures of 20th century molecular biology. He says, human intelligence, culture, and technology have left all other plant and animal species out of the competition. We also may legislate human behavior. In fact, governments invariably think that they can legislate human behavior. But we have too many illusions that we can, by writ, govern the remaining vital kingdoms, the microbes that remain our competitors of last resort for dominion of the planet. The bacteria and viruses know nothing of national sovereignties. In that natural evolutionary competition, there is no guarantee that we will find ourselves the survivor. Remember that the coronavirus breached all national, political, religious boundaries and had no respect for anybody or anything. Our virus is a legitimate subject of biology. They are, because they create so much of a problem. But are viruses a part of the kingdom of life? If you look at a biology textbook, they're not, because you have these three kingdoms of life: eukarya, bacteria, and archaea, and nothing else. The viruses don't find a place. So people in the literature do worry about this. Some people say there are 10 reasons to exclude viruses from the tree of life. Other people say, why should we not include viruses in the tree of life? Because they exchange genes across the super kingdoms of life. So this is something that those of you who are students might actually think about. Another way of saying this is, should viruses be studied by chemists, or should viruses be studied by biologists? Or is there any difference at all? And that brings me to the subject of my real presentation on nature's technologies and the evolution of biochemistry. I've put on this slide the images of Darwin and Mendeleev because they are the foundational pillars of chemistry and biology. Nothing in biology will make sense to you, and this is what Dobzhansky said many years ago, except in the light of evolution. And without Mendeleev, of course, we don't have the periodic table. Sadly, the periodic table lies usually unrecognized and dusty in the walls of chemistry lecture halls in many colleges. Today's technology day, and here's an example of a nature's technology with which we are trying to interfere, and that is the global carbon cycle. And look at the global carbon cycle. You see it in 12th class. Maybe not illustrated in the way I've illustrated it here, but you see it in 12th class. There's such an economy of operation here. What do plants do? They simply take carbon dioxide from the atmosphere, convert it into carbohydrate using energy. Energy is needed for anything chemical, and that energy comes from sunlight. So they're harvesting an inexhaustible source of energy, at least inexhaustible on a time scale of 5 billion years. After 5 billion years, the sun will be gone. And when the sun loses energy, we'll all be gone. But 5 billion is a long time away, so we don't worry about it. At this point in time, all you need to know is carbon dioxide goes in and carbohydrates produced. We consume the carbohydrates and we're happy. And what comes out? Oxygen comes out. What do the rest of the animals do? They breathe in this oxygen and they're happy. And they produce carbon dioxide again. And this is the global carbon cycle. And you can see, if you were a chemist, you'll see water coming out on both sides. It's important, because water coming out on both sides, then one of the things that you're actually doing in photosynthesis, you're splitting water into hydrogen and oxygen. And today, of course, the government's 20,000 crore green hydrogen mission would be a way of producing hydrogen. So you might ask the question, where did hydrogen go in biology? Hydrogen doesn't seem to be there anyway, it sort of disappeared. Where did it disappear? It disappeared as the proton. And what happened to hydrogen? Took a hydrogen atom, took a proton on one side, electron on one side, and that's what the mitochondrial electron transport chain does. And there it is. With this kind of complexity in biology, one might ask, what is the timetable of evolution? And this timetable of evolution is from a geologist, which I picked up from an article in Science Advances. The reference, I think, has disappeared down somewhere down below. But I've just annotated this. What is the time scale we're talking about? We're talking about the time scale of 4.6 billion years. 4.6 into 10 to the 9. It's very important, if you're a student of science, to think in powers of 10. Because you must think in powers of 10 in orders of magnitude, whether you're talking about length scales, energy scales, time scales, all these scales. In India, the only people who think about in powers of 10 are those who are doing scams. They think from lakhs to crores to so on, where the number of zeros keep on going up. But it turns out that students usually don't think in powers of 10, so you must think in powers of 10. Which are the first subjects which came to Earth, along with, at 4.6 billion, one hot spherical ball of molten metal, most inhospitable surroundings? This is the birthplace of biology. And you can't imagine biology when the Earth was born. There were only two subjects you should worry about then: geology and chemistry. You had chemistry all right, you had geology all right. And now geology and chemistry should somehow conspire to give us biochemistry. And it's only after we've got a little bit of biochemistry can we think of a cell. Can we think of a bacterial cell, however primitive, under very, very difficult conditions. So the evolution of microbiology must be preceded by the evolution of geology, the evolution of chemistry, and the evolution of biological molecules in biochemistry. Oxygen appears somewhat later. In order for oxygen to appear in the atmosphere, we have an anaerobic Earth. Bacteria must learn how to do photosynthesis. Plants came much later. The National Institute of Plant Genome Research must realize that on the time scale of evolution, land plants are actually long after animals. So that's what I'm trying to illustrate here. Microbiology precedes zoology, and zoology precedes botany. This is the hierarchy. Almost everything that we worry about in education today, medicine, engineering and all, they are in a dot on the extreme right of the scale, or not even the last couple of 100 years. That's the time scale over which we are looking at evolution. But we need technologies. If you're going to study anything, and we're going to study science, we need technologies. What are the technologies of science? I've illustrated two here. Early science is constrained only by one factor: by our eyes. We can try and explain what we see. We don't know what we can't see. Galileo's telescope and Leeuwenhoek's microscope are the ones which opened up two fields of science. Galileo goes upwards, cosmology. Leeuwenhoek goes downwards into a drop of tap water. He finds little bacteria swimming around. He's discovered microbiology. Two subjects of science come with these two technologies. So here I think it's very important to remember the British theoretical physicist's famous dictum. Freeman Dyson said this. And what he said was, science is often driven by new technologies rather than by new concepts. And there is no discipline that has been driven by new technologies more than modern biology. Everything in biology in the laboratory is a technology to the student. Sequencing, metabolomics, proteomics, genomics, PCR, anything and everything, even the poor green fluorescent protein, which sort of, and many other of its relatives, which drive cell biology, they've been forgotten as proteins. They're only known as colors. When you think about evolution, no one can think about it better than Darwin. And right early on, Darwin said there's grandeur in this view of life, with its several powers having been originally breathed by the Creator. He was a religious man, into a few forms or into one. Today, of course, if you think a lot about evolution, you will not be religious. But while this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Remember that when you look at the tree of life, we all are descendants of an ancestral cell. And therefore, everything on earth, biological, living, is descendants of an ancestral cell, and therefore we are all related. And in that sense, biology teaches you unity. And genomics also teaches you unity when you do comparative genomics. The underlying biochemistry is sometimes very much the same. And today, in the kind of climate in which we live, it is important to understand that unity extends across many, many barriers which we artificially create in biology. Monod, one of the great thinkers of the 20th century, of course, sometimes had a very depressing view of the future. But here that he says, living systems, there's no life below the level of a cell. So it's a single cell whose evolution that we are really worried about when we think about the evolution of biology. Cells are to biology what atoms are to chemistry. I studied chemistry, I drifted into biology, and therefore I think I would say this. Without atoms, you can't do any chemistry. And if you want to know the importance of atoms, wonderful statement made by Richard Feynman, probably the most quotable physicist of the 20th century. What he said in volume 1, first lecture, he says matter is made up of atoms. And he says if in some cataclysm, he asked the student this question, if some cataclysm, all of scientific knowledge were to be destroyed, and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? The cataclysm he's talking about is nuclear war, 1960s. And then he says, I believe it is the atomic hypothesis. And then he goes on to say, what the atomic hypothesis? So today, when we are on the threshold of a repetition of the 1960s, Mr. Putin on the one hand and Mr. Netanyahu on the other hand are threatening war on all sides, then of course you might wonder, okay, we have a war, we'll destroy everything, but then if you're very optimistic, we'll start all over again. And if we start all over again, what should we start with? I hope that some of us are left who will remember the atomic hypothesis. If Mendeleev's periodic table is iconic of chemistry, nothing is a greater symbol of biochemistry than the metabolic chart. I'm sure most of you have seen the metabolic chart, and I'm sure none of you have gone near it. It also hangs in the lab in many laboratories, and you can look at it from a distance. You can't read anything. It's just too many chemical reactions, all of them connected to one another. And these are what one might call the highways, the crossroads, the streets and bylanes of cellular chemistry. So you can see what will happen if you have a traffic jam on one of these. So that takes you then to the kinetics of the chemical reactions, to thermodynamics, how do fluxes happen in cells, and so on. It's important to know chemistry. It's important to know biochemistry. But what has happened with biotechnology? And here, Rajesh, I'm not blaming you. This cartoon sums it up. What's a 9-letter word for biotechnology? The man is solving a crossword puzzle. The very bright young lady sitting next to him says immediately, chemistry. And I would say that if you were asked a question now today, what is a 9-letter word for material science? Then you would again say chemistry. So it doesn't matter at which end of the application spectrum you're worried about, you must know your chemistry in order to get anywhere. But what has happened is that biotechnology and the promise of application has really washed away all the bridges to biochemistry. And this is why we today have an educational system in which you get degrees in biotechnology, but nobody can define what biotechnology is. But this slide, of course, tells you the most successful application of biotechnology thus far, and it's been there for 1,500 years. And this is the discovery of fermentation. And so the breweries were known long before. The breweries make a lot of money today, and the conversion of sucrose into ethyl alcohol and carbon dioxide is still a much studied process, a process which everybody tries to understand. It is precisely what lies behind fermentation, which is the starting point of biochemistry. That's what Hans Bruckner did when he crushed up yeast, filtered it through a funnel, and took the extract, no cells now, and then showed that it can do fermentation. That's how enzymes, biochemistry, everything was discovered. In this book of Arthur Kornberg's, he asks the question, how come Buchner, who was not a good chemist, how did he discover this and become the founding father of biochemistry, while neither Berzelius, nor Wohler, nor Liebig, nor Pasteur discovered this? Then he says, this is largely because some of them were immersed too much in chemistry, others were immersed too much in biology, and they didn't see the connection. But Buchner, who was a little bit ignorant of both chemistry and biology, really managed to make the difference. This is the historical timeline of biochemistry over the last 150 years. In some ways, it's also the historical timeline of biotechnology. From 1875 to 1925, a 50-year period, is when everybody hunted for microbes. This is completely, the history of this is described wonderfully in this book, The Microbe Hunters, which inspired generation of people to enter biochemistry and microbiology. Causes of infectious disease, microbial pathogens, Pasteur, Koch, immortal figures. Then came nutrition. We have so many diseases of nutrition. What caused those diseases? That's when the vitamins were discovered. Every vitamin which you think of was discovered then. But what was the purpose of the vitamins? What were they doing? Nobody knew anything about enzymes. They didn't know about coenzymes. So the fact that vitamins were coenzymes, facilitating biochemistry, facilitating cellular metabolism, came to be known only between 1950 and 1975. That's the period which I would call the enzyme hunters. This was the golden period of biochemistry. It is in 1953 that Arthur Kornberg discovered DNA polymerase. And without DNA polymerase, without Hargobind Khorana's synthetic oligonucleotides, which are the product of organic chemistry, none of what you know today in molecular biology would have been possible. The genetic code would have taken much longer to unravel. From 1975 to 2000, after Sanger sequencing came in, what were we doing? We were hunting for genes, gene by gene. And we were gene hunters. So we hunted microbes, we've hunted vitamins, we've hunted enzymes, we've hunted genes. Now, of course, after 2000, Venter and Collins now declared the human genome done. We now have genomics. But now, of course, what are we hunting for? And I suspect we don't know. That's why I've put a question mark there. That is from 2000 to 2025. We only have a few months to go. And in that case, I would leave it to the students. If you come up with a good idea of what we're hunting for now, maybe you can tell me, and I will put it onto my slide the next time I talk to an audience. Professor Shubhra Chakravarti took me to the metabolomics facility. Today we know every constituent of the cell. Here, abundant metabolites in exponentially growing E. coli. The hydrogen atom of biology. Every metabolite, every concentration. I don't think anybody looks at it. And if you look at it, you would wonder, look at what we have in abundance. Glutamate, wonderful, central molecule of metabolism, 96 millimolar. Glutathione maintains redox equilibrium, reduces you. Remember that aging is a process of oxidation, a sort of irreversible process which happens because of our exposure to oxygen, which we are breathing all the time, because oxygen is a toxic molecule. And there we are. These are the molecules. So as students, you might wonder, what are these molecules doing? They're all being used for a technological purpose. A technological purpose, very important, to keep the cell alive and growing. Chemistry must enter. So I put all the structures here because I know that in most biology audiences, nobody likes chemical structures. So if you now say, I'm going to find 5,000 molecules, then you must know 5,000 structures. If you don't know 5,000 structures, there's not much reason to find 5,000 molecules because you don't know what to do with them. And remember glutamate very much, because every time there's a furor that Maggi noodles has got monosodium glutamate, you might wonder, why is the government so excited about monosodium glutamate when we have so much glutamate already inside us? Here are the other molecules. These are molecules which you might like because they're made by plants. And it turns out that some years ago, I used to wonder, who are the best chemists? Microbes, animals, or plants? And I would always choose microbes. Now I've changed my view. I think the best chemists around are plants, and they make the most metabolites, and each metabolite more complicated than the other. I've put two metabolites here, morellin here and strychnine. Morellin, I put it there because I'm interested in morellin, because it's historically the first natural product whose structure was determined in India, and it was determined by none other than Professor G.N. Ramachandran and Gopinath Karta at the Indian Institute of Science in Bangalore many, many years ago. Strychnine, of course, all of you know. You don't like someone, you put a little bit of strychnine in their soup or their drink, and they're done. Vitamin B12. To me, it looks beautiful, but to most others, it would be a monstrosity. It's an organometallic. It's got cobalt in the middle. Nature makes this. Without B12, we won't be alive. We don't make B12. We don't make many things. We are parasites. Without B12, many critical metabolic reactions, which are very difficult to do, cannot be done, and only microbes make it. So in biosynthesis, you have two things. You have primary metabolites, cellular chemistry, and you have secondary metabolites, which is the biological imperative of an organism to make it. Why does the plant make strychnine? It makes it not for you and me, but it makes it for its own purpose. We should understand that purpose. That is chemical ecology. But look how strychnine is made. 22 enzymes. I put this up only for one reason. It's made by a plant. It's got 22 enzymes. That means it has got 22 genes. And therefore, you must clone all these genes and understand how the molecule is made. The strychnine synthesis, chemical synthesis, is very difficult to do, has been done. But most importantly, the reference, which is, you can barely read it. It's in Nature, and it's in 2022. So it's only very recently that this entire biosynthetic pathway has been worked out by a group at a Max Planck Institute. So biosynthesis is hard to do, and you need to do this to understand. Vitamin B12, 36 enzymes. All of them genes have been cloned. So you can see the organism is investing an enormous amount of energy, metabolic energy, in doing all of this. So it's doing it for its own purpose. This is technology in action. Today, if we were to produce these rare molecules in vitro in a system, using only enzymes, it is this kind of technology which one needs to understand. And you can see I've put these because I think nowadays people are more fascinated by the title of the journal in which a paper is published rather than the content of the paper itself. But sometimes if you look at these nice big journals, you do find papers which are relevant to whatever one is thinking about. But cellular metabolism is very complicated. And I think there's much recent work which is coming in which people are trying to understand the logic of cellular metabolism. Why do cells do things the way they do? How is energy really channeled in the cell? And what are the economies of this kind of metabolism? I like this slide very much because I've taken it from a review by Chris Walsh before he died, which says 8 kinetically stable but thermodynamically activated molecules that power cell metabolism. He's added oxygen there, but then you can see what the molecules are. You can see whether your favorite molecule is there or not. But I draw your attention to the fact that these molecules are kinetically stable but thermodynamically activated. That means they are reactive, yet they are stable. And it's only under specific conditions under the cell we can have all these wonderful transfer reactions that you can do unfavorable reactions using the energy that is stored in these molecules. But in addition, to get life, you need elements. And where do the elements come from? We think the elements come from Mendeleev's periodic table, but the elements actually come from the earth itself. There's nowhere else. Mendeleev's periodic table, if you're a student, please go and look at it. I like this because this book is written by a neurologist, Oliver Sacks, and I bought this book. It's called Uncle Tungsten, and he describes Mendeleev. He says he looked like a cross between Fagin and Svengali, with a huge mass of hair and beard and piercing hypnotic eyes. This is why I think today, if we wanted to have an evangelist for science, it might be good if we got Baba Ramdev or someone to be an evangelist for science. He might attract more attention than someone like me coming and wearing a nice clean shirt and telling you about science. He says a wild, extravagant, barbaric figure, but as romantic in his way as the Byronic Humphrey Davy. I spend hours now enchanted, totally absorbed, wandering, making discoveries in the enchanted garden of Mendeleev. I haven't found a better description of the periodic table than this. It is truly the enchanted garden of Mendeleev. But look at the enchanted garden of Mendeleev. Biology and life are dominated by carbon. Just below it, in the same column, you will find silicon. So one dominates the information age, the other dominates the age of biotechnology. Where do these elements come from? They come from the stars. All the elements on Earth were the Earth's inheritance when it was born. And Bronowski, in his famous series, The Ascent of Man, describes this process. He says, in all the stars that are going on processes which build up the atoms one by one into more complex structure. Then he says, matter itself evolves. The word comes from Darwin and biology, but it is the word that changed physics in my lifetime. Today, one of the sad things is that even biology students don't seem to really connect the word evolve and the term evolution. He then describes how a single carbon atom is formed. Very improbable collision, 3 helium nuclei at one point, happens in a billionth of a second, a millionth of a second. Then he says, every carbon atom in every living creature has been formed by such a wildly improbable collision. This should give you a respect for life, because life itself on this scale of planets is highly improbable. But when we look at biology, biology has evolved over time. Mendelian genetics and Darwinian evolution have been bridged by the chemistry of DNA. But just before they were bridged by the chemistry of DNA, Erwin Schrödinger, one of the founding fathers of quantum mechanics, actually worried about this. He worried about the nature of the gene in a set of famous lectures in the mid-1940s, just as the World War II was reaching its absolute peak. He was in Ireland at that time. And he says we call it in technical language a locus, or if we think of the hypothetical material structure underlying it, a gene. And then he wondered, how was this gene so stable that it was transmitted from generation to generation? When I read this, I was also puzzled. I said, where did this word gene actually come from? And I was asked by the Indian Academy of Agricultural Sciences to give a lecture. It was, I think, on a Mendel anniversary. I asked them, why do you ask me to give a lecture on Mendel? I'm not even a biologist. They said, doesn't matter, you can read and come. So I did read. So I went back. So I thought, having spent all this time reading, I might as well put it up. And you will find that the word gene first appears in the American Naturalist. This is a paper written by the famous Swedish Wilhelm Johansson. And therefore, he's talking about genetics. I rather like what he says about genetics. He says the science of genetics is in a transition period, becoming an exact science, just as chemistry in the terms of Lavoisier. Remember, Lavoisier made the balance and began to weigh everything. That's when chemistry became quantitative. The beginning of the 20th century, this is 1911. Johansson then decided that we need some more quantitative stuff in genetics, and there must be a unit of inheritance. And then he says, I propose the terms gene and genotype, and some further terms as phenotype and biotype to be used in the science of genetics. I was rather proud that I could discover this on the internet, because the internet is an inexhaustible source of information. But you must also remember that if an old man like me is able to discover this information, you should be able to do it much better. Schrödinger's questions were much more elaborate. He asked, how can events in space and time take place within the spatial boundaries of a living organism? And he answered his own question saying that Eventually, they will be understood by science. He talked about the problem of the second law of thermodynamics and entropy and the living cell. And of course, when he talked about entropy, he talked about Boltzmann. But then, things have progressed. Once Avery found DNA as the genetic material, and I hope most of you remember Avery before you remember Watson and Crick. And he's the one who did this. Go back and read Avery's paper in the Journal of Experimental Medicine. You will find how papers were written in those days. And Avery concluded, if the results of the present study of the chemical nature of the transforming principle are confirmed, then nucleic acids must be regarded as possessing biological specificity, the chemical basis of which is as yet ununderstood or undetermined. In honoring Avery and his discoveries, Lederberg, when he received the Nobel Prize in 1959 for his work in microbiology, he said that genetics should now be recognized as also timely for its axial role in the conceptual structure of biology and for its ripening yield for the theory and practice of medicine. And then he says experimental genetics is reaching its full powers in coalescence with biochemistry. And then he goes on to say how every phenotype must be described with the sequence of amino acids in a protein and so on. Then he said something which I think is very important, and I think this is what laboratories should recognize. He says the precise demarcation of genetics from biochemistry is already futile. But when genetics has been fully reduced to its molecular foundations, it may continue to serve in the same relation as thermodynamics to mechanics. This is the difference between physics education and biology education. In physics education, some things are fundamental. In biology education, nothing is fundamental. In biotechnology education has no foundation. One has to decide what the foundations are and teach them from the beginning. There are three pillars of modern biology: genetics and evolution. They are linked by chemistry. Remember, the Watson-Crick structure is nothing but hydrogen bonds holding two strands together. So the moment you come to hydrogen bonds, you are in the realm of chemistry. So on the timetable of evolution, this is what it is. Prokaryotic evolution, anaerobes, all the way till the evolution of oxygen. The invention of photosynthesis is very important 2.5 billion years ago. That's when cyanobacteria really began to learn how to do photosynthesis. And I won't go quickly into this because I'm coming to the end, but if you look at this scale, you will see that much of biochemistry must have already evolved. The Krebs cycle, the citric acid cycle must have evolved. Vitamin B12, that monstrous molecule that I showed you, must have already come. And the porphyrins, chlorophyll, heme, the pigment of blood, the green pigment of plants, they must also have come by abiotic synthesis. And then most importantly, energy. Is there anything that you need for life? It's energy. Now you will say, where is energy coming from? ATP. ATP, like DNA, biology has been reduced to two abbreviations, ATP and DNA. Every student will say this. And DNA now is, of course, adopted even by politicians. Only yesterday I was reading that someone said that DNA is in the communalism is in the DNA of the BJP. And someone else responded saying corruption is in the DNA of the Congress. So the idea that DNA sort of conveys inheritance is understood by common people. Sometimes scientists must understand energy and how energy now is used usefully. But before oxygen and after oxygen, biochemistry exploded. Eukaryotic cells could not have come without oxygen. And eukaryotic biochemistry vastly needs other things. I show you only 3 molecules here because I know that every biology audience don't like molecules. No molecules, you're not here. So you must be respectful of molecules. Cholesterol, collagen, and lignin. Collagen, of course, everywhere. Cholesterol, I tell you why it's important for the next slide. But lignin, no wood, no big plants and trees. If you don't have big plants and big trees, you can't harvest sunlight very well. No point in having a
sort of surface blanket of cyanobacteria, energy efficiencies will be very, very low. So in order for plant and animal life to thrive on the planet, in order for the Cambrian explosion to happen, you need the evolution of the land plants and have them grow. You need the tropical forest so that the harvesting can be very efficient. Collagen, of course, gives us no collagen, no animals. But we will teach collagen saying it's the most abundant animal protein. But it can't come without the hydroxylation of proline, and the hydroxylation of proline has to be done with molecular oxygen. But when I talk about this, remember, that's the structure of cholesterol. This is why structures are important. There's a single oxygen atom which I point with an arrow. And when he was doing his PhD, Konrad Bloch was asked this question: where did that oxygen atom come from? Nobody knew. He then decided to work on the biosynthesis of cholesterol. He worked it out. He received the Nobel Prize for this. And why is that important? Because it turns out that the cholesterol biosynthesis is a very complicated biosynthesis, but look at everything else that you get from cholesterol. You get the steroid hormones, you get the bile salts, you get vitamin D, so you need bone, you need the differentiation, you need the sex hormones, you need bile salts in the gallbladder, and then membrane fluidity is completely modulated by cholesterol. So it's a central molecule. And as a result, its biosynthesis is important. But its biosynthesis can't be done without oxygen in the atmosphere. So eukaryotic biochemistry and prokaryotic biochemistry differ after the evolution of oxygen. And here is the last, this is what everyone's searching for, the last universal common ancestor. Many of people here may not like it, but just to think of yourself as being descended from one single cell is quite a humbling thought. And of course, where did the eukaryotes come from? Lynn Margulis wrote this famous paper. Nobody will read it with a title like this nowadays, called On the Origin of Mitosing Cells. This is when she introduced the endosymbiosis theory. And this paper was rejected from, I guess, about 10 journals because before it was eventually published in the Journal of Theoretical Biology. Today, of course, it's a starting point for our thinking of the evolution of the higher organisms, when you go from prokaryotes to nucleated cells. I'm a chemist, so you must say, what are the starting materials for all this? This is all you had. Hydrogen, nitrogen, oxygen is a latecomer, water, carbon dioxide, iron and sulfur. And maybe with a little extraterrestrial help from the comets which were bombarding the Earth, hydrogen cyanide and formaldehyde. They are there in the comets. And you can take hydrogen cyanide and polymerize it, and you can get adenine and guanine, hypoxanthine, all of these molecules. Could have been a precursor for the nucleic acids. I'm coming to the end. The 3 pillars of modern biology I show you again. Ask you a question. What are life's imperatives? Survival and reproduction. And in fact, survival only until you reproduce. After that, you're useless. That's biology. What are the ingredients for life? It's information and energy. Where is the information? It's in the genome. It's in your institution's name itself. Everybody knows about information. Biology, to some extent, has been blinded by information, as we all are. All you have to do is look at social media or television or anything else. You are sort of completely swamped by information. Genomic information swarms all of us. Let's turn to energy. Very little understanding of energy. Energy, we have only Mitchell's ideas on chemiosmosis and how ATP is synthesized in the mitochondrion using a proton gradient, electrochemical gradient across the inner mitochondrial membrane. Remarkable process which happens with so many proteins, all of which are now being understood in great detail. And look at what there is. You need electron transfer. And there you have electron transfer, nicely separated iron sulfur clusters. Wonderful. And of course, St. George's famous quote I put up here, he says, life is nothing but an electron looking for a place to rest. Because you see, after we've stripped the electrons and protons, the protons push to one side of the membrane. What do we do with the electrons? We have to take the electrons somewhere. We can't have electrons like that. Eventually, the resting place for the electrons is the oxygen molecule. It reduces oxygen and it becomes water. Oxygen is the terminal electron acceptor in the respiratory chain. But if you look at those clusters are there even in nature. The mineral greigite has the same iron-sulfur cluster structure as what is found in proteins. And today's ideas of early evolution think of evolution of metabolism well in advance on structures which are completely inorganic, no lipid membrane, but really microporous materials which can be formed. And many wonderful experiments now are being done to show what these kind of structures can actually do. But chemiosmosis itself, which Mitchell introduced, is fundamental and universal because the archaea produce ATP the same way we produce ATP. Before oxygen, after oxygen, there's a different electron acceptor. But Mitchell himself was a very controversial figure. But now, in retrospect, after the 1970s, he did receive a Nobel Prize in 1978, which was rather controversial. But chemiosmosis and the idea of energy metabolism, or how ATP is produced, has been described as the most counterintuitive idea in biology since Darwin, and the only one to bear comparison with the ideas of Einstein, Heisenberg, and Schrödinger. See, biology does not have many counterintuitive ideas. Biology progresses slowly. The only counterintuitive idea in biology was Darwin, which is why he's still controversial. And Mitchell also is still controversial because this is how energy really is produced in the cell. This is Mitchell's original paper, which talks of many things that we might be interested in. He says he recognizes the biological membrane as critical, and he says we might be inclined to recognize that transport and metabolism, as usually understood by biochemists, may be conceived advantageously as different aspects of one and the same process of vectorial metabolism. And that is, I think, very important to understand the role of the biological membrane in making reactions go in one direction. Oxygen, subject of great interest nowadays in the general literature. The multiple books have come on oxygen, all titled Oxygen. So I would urge you, I put Lavoisier there as one of the three founders of oxygen. You can see there's a balance also there on the Lavoisier statue. Finally, a last word about evolution. Bronowski in his book says that among the multitude of animals which scamper, fly, burrow, and swim around us, man is the only one who is not locked into his environment. His imagination, reason, his emotional subtlety and toughness make it possible for him not to accept the environment, but to change it. And the series of inventions by which man from age to age has remade his environment is a different kind of evolution, not biological, but cultural evolution. I call that brilliant sequence of cultural peaks the ascent of man. But when you look at this, every cultural peak that Bronowski describes is based on an advance in science and an advance in technology. Whether it is the invention of fire, the wheel, the domestication of animals, the domestication of wild plants, the invention of agriculture, animal husbandry, and of course gunpowder, all of these are now, they shape human cultures, but they are all products of technology. And I think it's wise to remember them on a day in which we celebrate technology. But at the same time, it is important to also remember a little bit of cultural history. And this is in Yuval Harari's book, Sapiens, where he traces the history of humankind. He asks a real question. He says, once cultures appeared, they never cease to change and develop. And these unstoppable alterations are what we call history. So he asks, when did history begin? He says the cognitive revolution is accordingly the point when history declared its independence from biology. What I want to try and explain to you is the history of biology. Where did the molecules come from? How, that's what we have to think about. But what is the history that we think about? We think about the history of the last 1,000 years, the last 2,000 years, last 3,000 years, last 4,000 years. Not more than that. Very short period of time. And then he says from the cognitive revolution onward, that's when the human brain developed, he says to understand the rise of Christianity or the French Revolution, it is not enough to comprehend the interaction of genes, hormones, and organisms. It is necessary to take into account the interaction of ideas, images, and fantasies as well. Today, when we live in an age where mythology rules, you might actually ask this question. When did mythology appear? It appeared only after the human brain began to be able to think of these things. Before that, we were only animals as long as we were hunter-gatherers. There are still hunter-gatherers left in some remote areas like the Andaman Islands, very small groups of people. So on Technology Day, I have to ask I might ask the question, what is technology? I might go back and ask the question, what is science? If I'm asked the question, what is science? I will say science is the study of nature. Then, of course, if you tell a child that science is the study of nature, the next question is, what is nature? And so here's the definition of nature. This appears in volume 1, number 1 of the journal Nature. Thomas Huxley asked to write the editorial, 1869. He doesn't write the editorial. He translates from the German an essay written by the German poet von Goethe, where he says, nature, we are surrounded and embraced by her, powerless to separate ourselves from her, and powerless to penetrate beyond her. There can't be a better definition of nature. You're a part of it. Everything around you is a part of it. And therefore, whatever you study is then becomes the study of nature, and then it becomes the study of science. So those of you who are students, if you want a reading list, you might look at this. Bronowski's Ascent of Man, Harari's Sapiens, Nick Lane's The Vital Question. And the last book is, I rather liked it. I bought it originally, I wanted to give it as a present for a child, but I didn't give it as a present for a child because when I flipped through it in the airport, it was a little bit too complicated. It was, in fact, a book that I should be reading rather than the child. So I think something else sufficed, maybe chocolates or whatever. And the book is with me. It's called The Unstoppable Us. And this is written for children. And the question is this, how did humans take over the world? Today we will say their ability to invent technology. But this book doesn't come to that conclusion. This book will come to the conclusion that humans took over the world by one single ability, the ability to tell stories. And every religion is the product of a story. And it is religions and myths which keep people together, keep societies together, and allow cultures to grow and so forth. Otherwise, it is all biology. So that's what I wanted to tell you on Technology Day. I have only one acknowledgement here on my slide. These are the two institutions, the Indian Institute of Science on the top left, National Center for Biological Science at the bottom right. Both of these have provided me a home for decades now. The IISc for over 4 decades, and the NCBS, if I stay a little longer, would have provided me for another decade. And that's where I'm able to read and think about some of these problems. Thank you very much.
Thank you, sir, for such a captivating talk. I request my colleague, Dr. Alok Sinha, scientist NIPGR, to deliver the vote of thanks.
Good afternoon. It's my pleasure to present this vote of thanks at the end of this Technology Day today. And on behalf of entire NIPGR family and on my personal behalf, I would like to thank today's distinguished speaker, Professor Balaram, for giving this such an exhilarating talk I think it was like going back to your MSc days or BSc days where you're in a biochemistry class and being mesmerized by a professor's lecture and thinking that how little I still know and how much we have to read. So thank you very much for this talk, Professor Balaram. I think not only the young minds, but all present in this hall and those connected to us by online are motivated by this talk to read and these books. I think I'm glad that at least I have read one of them out of the 4 what you showed in the last. And I urge all my students and colleagues to go through and read these books and get benefit of this talk. So, sir, I thank you again from core of our heart that you agreed to give this talk today and enlightened all of us. At the same time, we would like to show our gratitude to the Secretary, DBT, and DG BRIC for agreeing to accept our invitation and be present today at NIPGR for this technology day. So your message has been taken very well. I think our students will be proud that they are in their first job and this is the PhD scholar positions and you will be going ahead in their life with many other jobs very soon. Thank you very much for that, sir. We also like to thank our director, Dr. Subra Chakravarti, for being always encouraging us and motivating us to excel in whatever we are doing, and also for bringing good fundings to support our research, what we are doing at NIPGR. I'd like to thank Professor Hema Balaram also for joining us today along with Professor Balaram, and it was very pleasure to have you here, ma'am, at NIPGR. I'd like to thank our neighboring directors of our neighboring institutes from ICGEB, Dr. Ramesh Sonti, who happened to also be former director of NIPGR, Dr. Devasis Mohanty from NII, to grace this occasion and being with us on this day. I'd like to thank all our guests from DBT, guests from the neighboring institutes and universities, and all those who are connected to us by online today for witnessing this wonderful talk by Professor Balaram. I'd like to thank my colleague, Dr. Pinky Agarwal, for conducting this today's program so smoothly. Thanks are due to my colleagues for being such a wonderful colleagues and coming together, making this harmony among all of us, all the administrative staff of NIPGR, the engineering sections, the technical staff, and last but not least, to all our dear students and our researchers for making this NIPGR what we are today. So thank you all for today's end, and I invite all the dignitaries to join us at Archives. And thank you all.